Sir, with due permission, can we start the session, sir?
Sure, madam.
Respected delegates, very good afternoon. And here is the last session of our 5 days virtual faculty development program on pharmaceutical innovations and protection strategies, organized by Seven Hills College of Pharmacy Autonomous in collaboration with CSIR, Institute of Minerals and Materials Technology, Bhubaneswar, India. And for this today's session, we have an eminent and renowned speaker in the field of pharmaceutical sciences. Sir is Dr. T.E.G.K. Murthy, sir. And sir is a professor and principal of Bapatla College of Pharmacy, Bapatla. And thank you so much on behalf of our management and our principal, Dr. Niranjan Babu, sir, for joining this today's session, sir. I welcome you cordially for this session. And to introduce the citations of sir, I now request Dr. Pushpakumari, madam, to read the citations. Madam, it's over to you. Thank you, ma'am.
Thank you, Dr. Sucharita. I am highly privileged to introduce the epitome of knowledge and a true inspiration in the field of pharmacy, Dr. T. Gopal Krishnamurthy, sir, Principal, Bapatla College of Pharmacy, Bapatla District. Sir was awarded Master's degree in Pharmaceutics specialization from Birla Institute of Technology, Ranchi, the capital of the Indian state of Jharkhand, with distinction. Sir did a doctoral degree in the field of controlled release formulations from JNTU, Hyderabad. Subsequently, he launched himself into the academic field as an assistant professor in Bapatla College of Pharmacy in the year 1995 and promoted to associate professor, and from which Sir was elevated to the present level of principal in 2006. He shared his research experience on the platforms of 50 national and international conferences, workshops, seminars, and symposia. At present, Sir is the coordinator of AP Academy of Sciences for the Guntur region, and he is also the Central Executive Committee member for Indian Pharmaceutical Association. Sir received fellowship from Association of Pharmacy and Biotechnology and is a recipient of Meritorious Teacher Award and Best Researcher Award from JNTU-K, Kakinada. Sir is acting as editorial board member for various journals and visited Uzbekistan to deliver guest lecturers to the students in their institution to abreast them about the latest developments in the field in the year 2016. And to his credit, Sir has many valuable stones in his crown, like 8 patents granted, 13 patents filed, 4 books published, and 2 complete research projects sponsored by AICTE, 5 conferences organized with AICTE grants, and 1 conference organized with DBT grants. And Sir guided 26 PhD scholars and 106 MPharmacy students. And Sir has 270 research papers published in peer-reviewed journals. Thank you for giving me this opportunity to give the citation of my own guru. Thank you very much. Over to you, Sucharita.
Thank you so much, madam. And I now request Dr. Murthy, sir, to take over the session, sir. Thank you so much, sir.
Okay, thank you, Sucharita, madam.
Thank you, sir.
For giving nice introduction, Pushpa, madam, and the coordinator, Dr. Sarvana Kumar, and principal of the institution, Dr. Niranjan Babu, and the faculty members who joined from various institutions. So thank you for giving me an opportunity to share some of my experiences with respect to innovations, all those things. So at the outset, I wish to congratulate all the faculty members for joining. Why I'm congratulating means you are showing your keen interest to learn something, because pharmacy profession is lifelong learner. Means we have to learn always in order to improve our knowledge. So in this connection, I wish to appreciate all of you for joining and showing commitment towards learning this subject. So once I will share the screen. Just one minute.
Sir, kindly share this screen, sir. Sir, excuse me, sir.
Outcome 100 by 55. Oh,
Sir, excuse me, sir. Sir, Murthy, sir.
Madam, whether the screen is visible?
Yes, sir. It is visible, sir.
Okay, thank you. So innovations, and everybody is doing some research kind of activity in order to perform, in order to identify some innovations as a part of your project work while you are a student in PG or PhD, and similarly while guiding your students. So basically, what is innovation? How it is differentiated from discovery? Because it should be interactive session. Always, I wish to prefer to have an interaction session. Can anybody talk about discovery, innovation, invention, whether all three are synonyms, or is there any small differentiating a boundary line in between these terms? Can anybody? Discovery, innovation, invention. Just all are close words, but there are some different. Say, repurposing of drug during COVID-19, everybody heard about repurposing. Of course, repurposing is a old term, but it became more popular during COVID-19. The drug was earlier known to produce some activity. Later, it was found to produce some other activity, or it is found to be useful for the disease management of another disorder or disease. So repurposing comes under invention, innovation, or discovery. Anybody? I don't think bad. Why he is asking questions like that? repurposing. Okay. Ethyl cellulose. Every pharmaceutics persons well know about ethyl cellulose. Ethyl cellulose is known to be a release retardant. If somebody came to find out that the ethyl cellulose can be used as a disintegrating agent in a particular form or a particular concentration, then it is considered as invention. Thalidomide was found to be useful for motion sickness. Later, it is used nowadays for the management of cancer. Initially, it was meant for some other purpose. Now it is using for some other reason. So such kind of changes are shifting from management of one disease to another disease can be considered as invention or innovation.
Sir, excuse me.
Yes, ma'am.
Sir, sorry for interruption. Kindly share in slide share mode, sir.
Okay.
Presentation mode, sir. Presentation mode. Click on presentation mode, sir. Sir, click on fourth icon, sir.
Click on
Fourth icon, sir. Down right corner, sir. Down, right corner, down, sir. Beside your battery percentage, sir. Yeah, next one, sir. Now click on that, sir.
Now is it okay?
Okay, sir. Perfect, sir. Continue, sir.
Okay, thank you for your suggestion and helping me.
Thank
Because
you.
during COVID-19, I was expert. Later, it was not practiced by me. Because of that, some inconveniences happened. So please excuse me for that.
No issues, sir.
Okay.
Continue, sir.
Okay, what is this pharmaceutical innovation? Pharmaceutical innovation requires novelty of effectiveness. So here there are two concepts. One is novelty and other one is effectiveness. So what is this novelty of effectiveness means? In order to treat hypertension, there may be several beta blockers like atenolol, and later some other drugs from beta blocker like metoprolol was discovered. So compared to the earlier atenolol, if metoprolol is novel, no doubt, because the drug is not there earlier. Later it was identified, so hence it can be considered as a novel drug. But what is effectiveness? So the new drug is also producing similar kind of activity compared to the earlier drug. It is not considered as novelty of effectiveness. What I mean to say in other words is, if some scientists in Seven Hills College of Pharmacy developed some sustained release formulation by using ethyl cellulose as polymer, and that article is published in a reputed journal. So the persons like me attracted with such kind of article, and here I am proceeding with same drug, same sustained release formulation by replacing or substituting ethyl cellulose with cellulose acetate. If I am also getting sustained release, which is on par with sustained release product developed by Seven Hills College of Pharmacy, then it is not considered as novelty of effectiveness. Because the release profile which is prepared by me is similar with the release profiles of already existing formulation. It may be a novel because earlier no formulation was reported with the polymer used by me, and hence it is novel. But it is not effective, as its effective is similar to earlier reported developed formulation or product. Hence, it cannot be considered as innovation. So innovation always leads novelty of effectiveness. So it should be better than existing or reported formulation or product or drug, whatever it may be. So a drug can be considered as a pharmaceutical innovation only if it meets otherwise unmet or inadequately met healthcare needs. Means, in order to treat a particular disease or disorder, we are having medicines. Let us take diabetes. In order to treat diabetes, we are having several drugs. If a patient is comfortable with the earlier drugs, means no side effects, no adverse drug reactions, and blood glucose is completely regulated. If the patient is comfortable with the existing therapy, existing drug, or existing drug delivery system, what is the need to go for a new kind of drugs or drug delivery systems? So innovation is required only if the existing drugs, existing drug delivery systems are having some shortfalls. If you are preparing a new drug or new drug delivery system to overcome the drawbacks elicited by the existing molecules, then only it can be considered as innovation. So innovation requires identification of shortfalls in the current therapy, current drug delivery system. And the pharmaceutical innovation is time dependent. What is time dependent means? Penicillin is not considered as innovation. Penicillin was innovation, while Alexander Fleming found out that penicillin. Whereas nowadays, we are having more powerful antibiotics compared with the earlier or the existing or still widely used penicillin. So now we cannot consider penicillin is an innovation. Once upon a time, it was considered as innovation. So with respect to time, the products may not be considered as innovative as it was gone. Surpassing existing levels of performance in terms of established efficacy, safety, or both would be considered as pharmaceutical innovations. So existing performance, if say ethyl cellulose is able to retard the release up to 10 hours, if you develop the formulation more than that, it is able to retain in the drug in the body, means mean residence time. The existing formulation is having a mean residence of 10 hours. If you extended the mean residence time of that formulation, then that can be considered because it is having retention time in the more period of time. Or antihistamines. An antihistamine is having a common side effect of sedation. If an antihistamine synthesized by you is free from that sedation, drowsiness, then that can be considered as innovation. Or a drug is having bioavailability of 50%. If you are going with a particular polymorphic form, or if you are suggesting a particular granule size of that drug so that the safety is established, safety is improved. Simple example, nitrofurantoin. As per BCS clause, it is having dissolution rate limited bioavailability and micronization improves bioavailability. But at the same time, micronized nitrofurantoin is likely to produce toxicity. So we cannot go for micronization because of toxicity. If you are able to retain the safety or bioavailability, then it can be considered as innovation. Or aspirin, which is likely to undergo hydrolysis. If again aspirin is again BCS class 2 drug, which is having poor solubility. By micronization, its surface area is improved, dissolution better, but it is going to undergo hydrolysis. If you are improving bioavailability or dissolution by maintaining the stability, then that can be considered as innovation. And limited scope for pharmaceutical innovation which existing therapies often relatively good outcomes. Means, so if already a drug delivery system is satisfying the patient community, then whatever you are preparing a new innovation, then it can be considered as just a competition, and it cannot be considered as a blockbuster because it is not going to improve any benefits. So this is generally what is innovation, how. So in this graph, you can have more. So what is this innovation is? In the x-axis, you can find here 0 and here 1. Means a patient is suffering with a particular disease. Sorry. So a patient is suffering with a particular disease or disorder, and that patient is not receiving a good drug, right drug delivery system, his life is not no more. Then everybody will strive to improve, to discover, to develop a new drug molecule so that the death can be stopped. And some diseases, whatever we are having existing treatment, still the patient is not going to receive a drug or drug delivery system, he can survive. So there are two extremes here. One is the patient is suffering with some disorder, his life will continue as such without any deterioration, even no drug is available. And second case is a patient is suffering with a disease or disorder, until unless a good drug is not provided to him, the patient life will be in risk. Can you give an example for this? Means the existing therapy is there. With that existing therapy, patient is not going to be improved. The same condition will be sustained. In second case, the existing therapy is provided to the patient. Even then, the patient life will be threat. Means the present therapy is not at all extend the life. Just the patient may feel some comfort. So a gravity of unmet need means the patient needs a drug. Existing therapy is not at all suited to improve his health. But in one case, the patient will be in healthy condition, means he will be in live condition only, whereas in second case, the patient will going to lose his vital life within a short period of time. So in first case, I can give example of blindness. In case of blindness, the drugs are not available, or color blindness. So the patient life is not in threat. But if any drug is discovered, his eyesight vision may be improved, but life is not threat. Whereas in second case, a person is in fourth phase of cancer. The existing therapies are not able to extend the life. So that is gravity of unmet need. So innovation is required for both the purposes. Because in both the cases, the present therapy is not at all sufficient. So innovation is required in both the cases. In first case, the innovation may not grasp the market. Whereas in second case, because the patient life is in danger, so it may become a breakthrough in the market. It will receive a good attraction. So while innovation, while any person is showing interest in innovation, you have to identify what is the present scenario, what is the need for this innovation, what is the percentage of unmet need, and how to highlight it, how to exploit the situation. So that is gravity of unmet need. And in y-axis, comparative effectiveness. Comparative effectiveness means compared with the existing therapy, existing drug delivery system, how your innovation is superior, better with respect to the existing therapies. That is comparative effectiveness. So here there are three kinds of categories: incremental, substantial, and breakthrough. So incremental means just a slow bit of improvement with respect to your innovation compared with the earlier products or earlier therapies. Substantial, it is an intermediate between breakthrough, because in case of incremental also, if you are observing, it is going to fulfill up to 50 or 60%. Whereas in case of substantial and breakthrough, the chances of effectiveness is going to be increased. So this year effectiveness may be up to 40%. Whereas in case of substantial, it may be even up to 80. Whereas in case of breakthrough, you can have a more than around 100% effectiveness compared with the existing formulations. So that kind of things we have to identify while proceeding for innovations. Okay, meanwhile, anybody, can you try to clarify one doubt? Whether innovation is accidental or it is a scientific background? So innovations may be accidental or innovation may be scientific. Or some cases, it may be scientific accidental. So what is this accidental? Accidental is innovation by Newton's is one of the classical example. So this is discovery. Now that is existing phenomena only, falling of a fruit from a tree onto the soil. But actually, it is a discovery. So discovery is by accidental. And examples of innovations by accidental is the first artificial sweetener, the saccharin, is discovered without any scientific invention or interest. So it is purely accidentally it is discovered. And similarly, the world's first antibiotic, penicillin, is also by accidental by the scientist Alexander Fleming. And blood thinner warfarin is also an innovation identified by accidental only. Quinine, the persons who are suffering with malaria was accidentally taken this cinchona bark by considering it as a poison. But fortunately, they were all free from the disease or disorder, malaria. And thereafter, they identified systematically what is the active principle responsible to cure malaria from that bark, and that alkaloid is identified. Later, it was found to be a proven material to safeguard the human health against malaria. So whether my audio slides are visual, everything is clear?
Yes, sir.
Okay. So now the other one is innovations by technical or with a keen objective of a scientifically based hard work. Example, innovations by technology. The microscope is a technologically developed product. Polio vaccine is also not by accidental. The scientists done several works in order to complete development of polio vaccine and DNA sequencing and the current scenario 3D printing of pharmaceutical formulations in order to meet several objectives. So one of the advancing field in pharmaceutical product development nowadays is 3D printing. Still, there is a scope to do a lot of innovations on 3D printing in order to develop various solid dosage forms, semi-solid dosage forms, liquid dosage forms, controlled release formulations, utilization of machine learning, artificial intelligence, in order to patient-specific drugs or drug delivery systems, on-demand systems. So such kind of innovations are by means of technology only. Then why, before discussing about mechanistic models, why we can't go for mechanistic models? Why the traditional design of experiments, DOE? So you neglect the concept of the discovery or innovation by accidental. We are all technical people and try to do, and we are all in a platform to develop innovations based on our knowledge, that may be a scientific or technical knowledge. So we can restrict always towards the technical innovations rather than innovations by accidental. So in order to do some innovations, the better choice is let us do some experiments. And in order to do experiments, the traditional one factor at a time is not preferable nowadays, because the outcome is influenced by several inputs or in-process variables. So we have to go for multivariable analysis, as the response is triggered by several in-process variables. We have to control all in-process variables at a time. So better solution for that is design of experiments. And as design of experiments is available, why we have to go for mechanistic models? So in order to brief about mechanistic models, we should know what are the lapses or what is the need to go for mechanistic model based on just a fundamental understanding of the traditional DOE, design of experiments. So in case of DOE, there should be a proper planning to do experiments, as DOE can be performed by using some softwares. But in order to exercise that software, in order to give some matrix of experimental conditions, the input should be given by the man. So the human being should identify the factors or parameters which are likely to influence the response or outcome. For instance, a pharmaceutical scientist is showing a keen interest in order to minimize the disintegration time. So the response is disintegration time. And in order to alter the disintegration time, there may be several CMA, critical material attributes, critical process parameters. So one has to identify which material attribute is a crucial factor or critical factor in order to identify the disintegration time. So the number of factors influencing your response. Later, we have to fix the design space, at what range the identified factor or the factor of interest is influencing your response. So based on that factor and fixing the level, we can fix the design. So in order to go for whether factorial design or response surface design or mixture design or Taguchi design, because commonly in DOE, these four are the designs used. In all four these designs, you have to identify what are the factors or variables of interest and which factors we are going to control. That is called as blocking factors, because we are not going to, your study may not consider all the factors. We want to ignore some factors, and those are called as blocking factors. And Once the design, the factors and the levels are fixed, then we will be having a design matrix. And from that design matrix, we have to go for replications, means each experiment, how many times you want to perform in order to showcase the consistency, validation of your experimental results. Randomization means localized control, like in case of dissolution, how much volume of dissolution fluid you are going to use, what is the effect of dissolution medium temperature. So in order to keep that, we are having 2 more vessels where the dose is not going to be kept, but for replenishment of dissolution fluid, we are going to withdraw the specified fluid from that remaining 2 bowels in order to keep the temperature constant. So that is the randomization, means order of experiments, how we are going to perform the experiments, means each experiment will be having equal opportunity. Whether we have to do the experiment 1 with 2% binder should be the first experiment, or 4% binder should be the first experiment. That should be decided again by DOE software. It is going to give order of experiments, run order. So that should be traditionally observed in case of DOE. And in case of DOE, you will be having results. As per this protocol, you are performing the experiments. There will be a result. And from that result, you can compare a particular formulation is better compared with the other formulations. Say, one of the faculty member may guiding a student in order to develop a RPHPLC method where theoretical plates, retention time may be the crucial factor. They may be using different mobile phases. And one of the variable is a percentage istonitrile, minus percentage organic solvent. So as the percentage organic solvent is increased, or in other words, the retention time is influenced by percentage of organic phase. That is the conclusion based on that result. But how this organic phase is influencing retention time? A pharmaceutical scientist is prepared a formulation, SR formulation, by using 4 polymers, ethyl cellulose, cellulose acetate, erythrozyte, HPMC, etc. And after preparing the 4 formulations with the different polymers, they may be subjected to release studies. Based on the experimental results, they will identify a particular formulation prepared with a particular polymer is better. That is illustration or interpretation of the experimental result. But such kind of results is not going to give any idea about why that formulation is better, or why that polymer, why that excipient is superior compared with the other. And in order to address such kind of issues, mechanistic models are required. So in case of design of experiments, the data will be not be correlated with in order to identify the factor mechanism, how it is going to influence, alter the response. So if you identify the mechanism, then we can have several advantages. The one is sensitivity analysis. So sensitivity analysis in the sense, in case of this traditional DOE experiments, a simple experiment, particle size versus dissolution rate. As expected, particle size is reduced, dissolution rate is increased. But it cannot be generalized. That is applicable only in case of BCS class 2. That kind of knowledge is not obtained from the traditional DOE experiments. Because DOE suggests there is a relation between particle size versus dissolution rate. So you go for reduction of particle size. Whereas in case of mechanistic model, we can identify this is because of so-and-so parameter. And this parameter is applicable only particular classes of drugs. Whether a drug is having pH-dependent solubility, pH-dependent dissolution, pH-dependent stability, pH-dependent absorption, that cannot be considered, that cannot be identified in traditional DOE experiments. So we have to go for, so which parameter is sensitive? even minute fluctuations in that parameter, there will be drastic changes in the experimental results. So which parameter is more significant, which can be ranked as first, followed by which factor is crucial. So we can identify the parameters. We can rank them accordingly so that we can manipulate, we can deliberately change the factors or its inputs in order to get the required outcome. So that is the advantages of mechanistic model. And based on that knowledge of mechanistic model, we can go for physiological based models, or we can go for simulations, and that is possible only in case of mechanistic models. So in case of mechanistic model, in case of earlier, we can say by administering a drug, it is having, if you are suffering with some pain, you are taking drug, we can observe, yes, by taking this drug, my pain is reduced. And we can identify onset of action, duration of action. So after taking this drug or drug delivery system, in pharmacokinetic parameters like Cmax, Tmax can be observed. Based on that Cmax, we can say this formulation or this drug is having higher Cmax compared with that. But how it is going to produce its activity? What is the target? What is the receptor? How it is going to act? So in case of antidiabetic drug, by giving the drug, we can observe blood glucose levels. Based on that, we can have a knowledge that this drug is having antidiabetic activity. As after taking this drug, the blood glucose levels are compared to be less. Before and after treatment with the drug, there is a percentage reduction of blood glucose level, and we can say it is antidiabetic drug. But how it is producing lowering of blood glucose level? Because in order to produce the blood glucose reduction, there may be different mechanisms, and the drugs may act by different mechanisms. So in order to identify potential interactions between the drugs, or in order to identify a synergistic effect, additive effect, we have to know mechanism of action. That may be ADME interaction or pharmacokinetic interaction, pharmacodynamic interaction, drug-drug interaction, drug-food interaction, drug and beverages interaction. So in order to identify such kind of that, we have to identify the target receptor, where it is going to interact. And that is not possible with respect to DOE. So in case of mechanistic model, we can identify the mechanism. So how this formulation is going to act, how an excipient is going to act. So what is the reaction between the drug and excipient or drug and body, and that can be identified. Based on that, the mechanism, if it is known, we can go for simulation so that instead of doing a costly clinical trials, which are not only expensive, they are also risk, and they are going to be involved in the safety of the human being. So the regulatory agencies may object in some cases, especially those drugs which are having highly potent cytotoxic drugs. So in such cases, the regulatory agencies nowadays are allowing virtual clinical trials. In order to develop virtual clinical trials, you should know mechanisms, how it is going to act. And by knowing that, we can artificially, outside the body, we can create that kind of environment. So in case of absorption, there is no need to go for absorption studies on human being. The cells which are involved in absorption of a drug in the human being, by knowing the mechanism, which kind of cells are involved. By knowing that, we can artificially create cell lines. Nowadays, cell line cultures are going to be used in order to cultivate, in order to develop several studies, including absorption prediction studies, and similarly, in order to study.
అదే మీరు సండే రాస్తారా?
In order to identify how a drug is going to cure the cancer, whether this drug is having better activity compared with the existing anti-neoplastic drugs or not. So in order, because anti-cancer drugs are not safe, such kind of drugs can be performed by virtual clinical trials, which is possible with mechanistic models only. And nowadays, the new concept is personalized medicine. There are some patients may be highly reactive with a particular drug. As in some persons, metformin may be better. In some persons, glimepiride, some persons, pioglitazone, some persons, another drug. Or in case of antihypertension also, the hypertension management also, some persons may be better choice is AC inhibitor, some persons beta blocker. Again, in case of beta blocker, some persons may be responsive towards the old drug etenolol, and some persons may be better with the administration of metoprolol. Why it is so? Why the same drug is not going to fulfill the needs of the patient? Because it is not meant to treat all people. The one persons may have maybe better cured, or one person may be noticed some untoward incident with the administration of the drug. Why it is so? Why the person's behavior or response is different from one drug to another drug? So in order to identify such kind of incidences, and in order to prefer, in order to recommend a particular drug or drug delivery system to the patient, in order to safeguard the health of such kind of individuals, the mechanistic models might be useful. So that is why mechanistic models are more preferable for innovation compared with the traditional models. So a mechanistic model is based on theory. In case of dissolution, say Navier-Whitney equation is one of the mechanistic model because it is based on theory, like particle size or surface area, the concentration gradient, thickness of the barrier or diffusion membrane. Similarly, the absorption, in order to explain the proceedings of absorption of a drug, there may be a CAT model or ASCAT model or CETA model. So different kinds of models are developed in order to study the absorption or other ADME characteristics of a drug. So with respect to the stability of a drug, in order to explain dissolution, stability, solubility, there are different theories. And of course, the theories may also vary with respect to time, and some factors may be new incorporated, some factors in the earlier theory may be deleted. So that is all based on mechanistic models. So mechanistic models are based on some theory. We have to correlate earlier findings also, same scenario is observed, and here also we observed same kind of results. In such a way, we can correlate our findings with the earlier findings. So a sufficient or a background of good theoretical knowledge is required in order to develop, in order to establish a good mechanistic models. So mechanistic models are preferable only if you are having a good data. When sufficient data and the data is genuine, authentic, reliable, then only based on that data we can develop a good model. Simple example, a student may ask the faculty members like us that why we have to take 5 points in case of construction of calibration curve, like 2, 4, 6, 8, 10 microgram per ml, and we are taking absorbance of 5 concentrations. Why we can't go for only 2, that is low concentration and high concentration? Or similarly, in case of a dissolution, why we have to collect the samples at regular intervals? Why not the initial and the last point? Similarly, if a drug is given to a patient, why we have to observe the patient response at periodical intervals? Why not after 24 hours? Why at regular intervals like 4 hours, 6 hours? Why we have to observe the patient conditions? Because in order to fit a good model, the number of points, because a straight line drawn between concentration and absorbance, the line pattern is going to be different with respect to number of points utilized by the scientist. So good data is required in order to predict the model. Then only the model will be having good prediction and we can go for validation of such kind of models. And mechanistic models are also required to understand the process involved or to identify the response involved with respect to a drug, and we can monitor the process by nowadays we are having automation in process. In order to implement automation, we should know what are the parameters to be controlled. And in order to identify the parameters, mechanistic models are required. And this can be applied. Mechanistic models or the data or the results obtained from mechanistic models can be applied either by offline as well as online. In case of online, we can go for the process control. Say, in the sense, dissolution apparatus. Your objective is the dissolution process should be controlled for a period of 10 hours. And it is not possible to observe the temperature throughout the 10 hours manually. So we have to go for automation. And during the 10 hours, we have to control the temperature, RPM, and how to control this temperature, stable 37 degrees centigrade. If its temperature is reduced below 37, we have to set the heater in on position. If the temperature is rising more than 37, then heater should be set to off position. So how it is going to be regulated? So there should be a sensors which are going to measure the temperature in the dissolution bubble. If there is any fluctuations in the set temperature, then they are going to send signals, and based on that signals, the heater will be controlled. That will be automation. Similarly, in case of a tablet compression machine, the upper punch, the distance traveled by upper punch into the die cavity, the distance raised by lower punch while raising the tablet after its compression, the force exerted by the upper punch while entering into the compression force, they're all going to be regulated by online monitors, that is transducer gauges. So in order to set for automation, mechanistic models are better or so useful. For example, this is one of the illustration done by our students in order to establish some mechanistic model to correlate the solubility. And here we use some propionic acid derivatives of NSAIDs. There are 7 drugs: ibuprofen, naproxen, phenylphenoxy, ketoprofen, chlorpheniramine, etc. And these drugs solubility is experimentally determined. So in order to develop or construct a model, there should be a response. And here in this example, the response is solubility, and other one is the parameters which are associated with the drug. The parameters associated with the drug, we used some molecular descriptors like melting point, molecular weight, heavy atoms, aromatic heavy atoms, fractional CSP3, rotatable bonds, hydrogen bond acceptors, hydrogen bond donors, molar refractivity, topological polar surface area. These are all the properties of the drug, and these properties of these drugs we collected from the data source, that is Swiss ADME. In Swiss ADME, we entered the structure of these drugs in the SMILE string form. So that SMILE string we obtained for all these drugs again by using a data source, PubChem. So in PubChem, we collected this SMILE string for all these selected propionic acid derivatives. And while inserting this drug SMILE string in the another Swiss ADME software, we got all these properties: melting point, molecular weight, heavy atoms, atomic. So up to this TPS, topological polar surface area, we got all these. So these are all the values based on the arrangement of atoms on all these drugs. Another one is experimental solubility. So that is log S values. And these log values of these drugs were collected, and this is actually experimentally determined. Log solubility, this is not predicted. So log S experimental value, because always in order to develop a model, there should be experimental values required. And experimental log solubility is obtained from drug bank. So online drug bank, again, it is a free software. So log S values are obtained from online a book, that is drug bank. And this is from software, a free software, Swiss ADME. And from Swiss ADME, we collected all these properties. So one by one, so whether melting point is having relation with the log solubility, whether molecular weight is having a relation with the log solubility. Because in order to develop modeling, we should know relation, whether these factors are having relation in log solubility or not. So each and every parameter and its relation with the experimentally determined solubility, we analyzed by using another software, Minitab. So in case of Minitab, we observed the R-square value, correlation coefficient value of different properties, whether it is a linear relation, cubic or quadratic. And these are all R-square values. And the values obtained from different drugs is subjected to ANOVA. And ANOVA p-values are also depicted here. And we observed some of the parameters which are significant by set the p-values less than 0.05. Means what I mean to say is the factor which is showing an ANOVA p-value less than 0.05 is showing interest or showing its influence on the parameter of interest log solubility. So in this case, we observe molecular weight. So in case of molecular weight, in case of quadratic, it is having 0.03. Means molecular weight is influencing solubility and it is following a quadratic equation. Quadratic equation is y is equal to ax cube plus bx square plus cx plus d. And in case of cubic, the equation is y is equal to ax square plus bx plus c. And in case of linear, the equation is y is equal to a plus bx. So it is showing molecular weight is showing its importance or its effect on solubility. And the other one is the heavy atoms. In case of heavy atoms also, this p-value is 0.03 and other one is rotatable bonds. And in case of rotatable bonds, 0.017 is the observed p-value and R-square value 98.3. And the other one is molar refractivity. And in case of molar refractivity, again it is also following 96.6 R-square value and p-value 0.03. So molar refraction is also showing its importance on solubility. So based on this, we can say the log solubility of propionic acid derivatives can be correlated with the significant factors by using these equations. So this is mechanistic model, how a solubility of propionic acid derivatives are influenced. So the solubility of propionic acid derivatives are influenced by parameters like molecular weight, heavy atoms, rotatable bonds, molar refraction. And this equation we cannot generalize because the properties of other NSAIDs or other categories of other drugs which are used in order to manage other diseases or disorders may be different. So the mechanistic models can be developed. So based on this, how we can go for innovations? In order to get the required solubility, what should be the number of heavy atoms required? So here in this equation, we observe that heavy atoms are going to reduce the solubility because it is here, it is having negative sign. So by reducing the number of heavy atoms, we can improve this log solubility. So your innovation should be, your strategy for your innovation will be discovery or development of a new molecule with less number of heavy atoms, or go with a new compound having more rotatable bonds, because rotatable bonds is having positive effect. By increasing rotatable bonds, the solubility is going to be increased. And molar refraction, by reducing the molar refraction of a new compound, that is our innovation. So in order to go for some strategy for your innovation, some theoretical background is required, and that background is attributed by such kind of mechanistic models. And the other mechanistic model for same propionic acid derivatives is correlating the log p values. Again, these properties are obtained from Swiss ADME by inserting the smile string format of propionic acid derivatives, and we develop the relationship between the partition coefficient, because the lipophilic nature is one of the parameters required for intestinal permeability. Alternatively, okay, then I will have to. So this partition coefficient here, if you are observing, the p-value is found to be more in case of topological polar surface area. So topological polar surface area, here it is following cubic, whereas in case of solubility, it is showing quadratic equation, and in case of NO, it is showing, in case of log P, it is found to be dependent on topological polar surface area, and the topological polar surface area is showing a correlation coefficient value of 0.98 and ANOVA value is 0.03. So it is found to be influencing the permeability because lipophilic molecules are having more permeability. So lipophilic nature log partition coefficient. Again, these log P values are obtained from experimental values from drug bank. And it is following cubic equation. So mechanistic model is developed in between log P versus topological polar surface area. By using this, we can go for some novel compounds in order to predict the log P. So log P value can be predicted by introducing the TPSC value of a new compound belonging to same category. So that is possible with respect to the permeability. Alternatively, we can also go for CACO2 self-permeability values. If experimentally determined CACO2 permeability values are available, then we can predict intestinal permeability by correlating CACO2 cell permeability values with these properties. So by correlating this, we can identify which property of that drug is crucial in order to have good solubility, good permeability, good absorption, or by altering distribution, metabolism. So computational modeling is also possible by addressing the mechanistic knowledge or mechanistic models. So these mechanistic models may also be useful in order to develop the new drug or new drug delivery system in order to address a pathological conditions. Because a normal human being tissue organ is quite different compared with a person suffering with a liver disease, kidney disease, or heart problem, or the persons who are having some metabolic disorders. So how to simulate such kind of, create such kind of situations? In order to address such kind of issues, again, we can go for biosimulations. So mechanistic models are useful for biosimulations. And in case of biosimulations, there will be 3 steps: pre-programming, programming, and post-programming. In case of pre-programming, we have to first define what is our purpose, what is your innovation, and for which innovation you want to develop mechanistic model, and identify unit of interest. Unit of interest means which organ, which tissue, which cell, or which intracellular components you want to highlight. Even that may be cell, enzyme, gene, genetic expressions, such kind of things which you are interested, which kind of cell line cultures you want. So that is unit of interest. Identify system knowledge, because some cases in order to process your data into, that may be the data should be processed in order to expedite the process, in order to interpret your results. Which type of programming is required? Whether you want to develop a software or you want to develop a program with Java, Python, C, C++, etc. So which one is better suited for your, that we have to identify. And in case of programming, model population. So most of the cases, our PK/PD studies are restricted to a specific people. And specific people in the sense, a simple logic is, if you predict a pharmacokinetic parameters of adult male, whatever Cmax, Tmax, Ka, AUC, such kind of parameters generated, the data cannot be extrapolated to all human beings. Because the data which is used, which is with you, is only for adults, that also male. And adult male is quite different from adult female. that may be based on physiological differences or in some cases pathological differences. A person who is having renal impairment, then there should be a need to go for corrections or adjustment of either dose or dosing frequency. And a patient may have 20% kidney damage and another patient, the kidney may be damaged to 60%. Now the question will arise. The amount of drug to be given to 20% damaged kidney patient and 60% kidney damaged patient is same, or if it should be modified, then how to modify? So we have to create which parameter from that person can be correlated in order to identify the progress of disease or progress of disorder. Which biomarker is better to correlate the condition of the patient, that may be disease condition, or with the treatment of your drug or drug delivery system, the response module of the person, which biomarkers are better. So in order to identify that, we have to go for the parameters. And always these parameters will be estimated by experiments only. And experimentally determined parameters can be extrapolated so that it can be applicable for all population. That is considered as population pharmacokinetics. So how to extrapolate your knowledge from sample pharmacokinetics to population pharmacokinetics? How sample parameters are differ from population parameters? How to compute population parameters from sample parameters? And in order to transfer sample parameters to population parameters, different techniques are reported in the literature. Among those techniques, which technique is better suited for your work? That all you have to identify, and that should be fitted into a proper model. That is model development. Once this model is developed, that should be verified in order to identify the errors in that model. But what is this error means? Predicted response and actual response. The difference between the predicted and actual or observed response. If it is less, standard error is less, then you can say, yes, your model is perfect and there is no need to alter your model. If the predicted values from the model are quite different from experimentally observed results, then you have to reshuffle your model by deleting some parameters or by adding some parameters. Again, you go for revalidation of your model, and that process should be continued until you will get a validated model. Validated model is a model where the differences between the predicted values of your proposed model and actual or true values difference is quite less. So in order to address such kind of biosimilarities, biosimilars, biorelevant dissolution media, or simulation of human biology, mechanistic models are required. So that is one thing. And the second one is, these mechanistic models are useful for the development of cellular networks, which includes genomics and proteomics. So the advanced drug delivery systems will be completely utilization of the biotechnology based formulations. Even now we are all enjoying the vaccines, how they are going to act, and what are the shortfalls of present immunological products, how to overcome those hurdles or barriers, and in the management of cancer, how the cell proliferations, the cell surface structure, and identification, development of potential ligands to bind the receptors, to control the expressions of genetic material. There should be a model which is identifying the genes network or protein network. So in our body, we are having different kinds of networks like organ network. And we have to appreciate the Almighty or God. Compared with the human being, some cases one human being may not help the another human being who need help. Whereas in our case, if one organ is damaged, the function of that organ is shared by another organ. Say a drug is likely to be metabolized by most of the cases liver to develop a hydrophilic metabolite, subsequently which can be excreted. If liver is damaged, that part taken care by other organ, so that it will take the responsibility to go for metabolism. That may be kidney, that may be lungs, where the drugs may be excreted by such kind of other routes also. Or even in case of liver, the same drug may be metabolized by different enzymes, say ibuprofen, the propionic acid derivative, may be metabolized by cytochrome P450-1A2. Similarly, cytochrome P450-6A. Same ibuprofen may also be metabolized by glucuronide transferase. In a person, there is a gap of the secretion of a glucuronide transferase. Then glucuronide conjugation may be impaired. Then glycine conjugation may be improved so that the drug can be excreted. So that is sharing of responsibility. How that sharing is possible? Because of a strong network among the organs, among the cells, or among the genes. So how they are going to be interrelated? If that chain is known, we can utilize, we can exploit that chain for development of new chemical entities to safeguard the human health. And in our body, there are different kinds of networks, like interaction networks. So regulatory network, signaling network, metabolic networks. So metabolic network is simple example is if liver is not functioning properly, then food is not metabolized. So it is going to give signals to our body that don't take food or reduce the quantity of food today. So that is metabolic network. Actually, one organ is suffering, but it is transforming that signal to another organ which is involved in consumption of the food. And signaling network, say one mosquito bite if you are facing. Mosquito bite is observed in particular area, like it may be a leg. Then it is going to provide a signal to another part of our body, that may be hand or palm, so that it can be used to kill mosquito from the affected area. So such kind of signals. So which area is affected and how to provide a drug to reach that target. So identification of a target and what identification of barriers involved in transportation of the drug from site of administration to site of utilization, site of activity. Such kind of signals are useful. And especially in the cancer management, the control or regulation of network is more important. So metabolism should be, or cell proliferations should be regulated. So in order to regulate that material, which genetic sequence is required, how to increase the ability, how to block the other precursors involved. Because in our body, we are having a regulating mechanism for the stimulation of cell production and as well as destruction of the cells. So always there will be a driving force and resistance in order to balance the things. So how to balance the number of cells, cell growth, cell activity, which one should be driving force should be increased, and which we have to resist, which we have to regulate or control, how to simulate. So such kind of knowledge is possible with respect to mechanistic modeling. And so high predictive model of mechanistic model is possible only if you are having a sound scientific knowledge in order to identify a set of interactions between drug and body, drug delivery system and body, or even in development of a drug delivery system, what are the key excipients required, how to alter the release, how it is going to alter the release. So such kind of interactions are always required in order to develop a good drug or drug delivery system. And the parameters which are involved in mechanistic model, the estimation of the parameters should be in a scientific background with good precision and accuracy. Because the data is good, then only the final quality of that model is good. So if the parameters are not correct, then model will fail. So always ensure that the parameters which are used to construct the model should be established, should be measured under a scientifically well-designed experiment. That is the key area. So my suggestion to all the faculty members who joined this session to go for innovations. Of course, you are all involved in innovations, but try to improve the innovations based on good theoretical knowledge with appropriate mechanistic models. That is my sincere suggestion for you. And at this outset, I wish to thank all the faculty members who joined this session. And once again, I wish to thank the organizing committee for giving me an opportunity to discuss some insights about mechanistic models, how they are useful in establishing innovations and all those things. So feel free if you are having any queries with respect to these mechanistic models, I will try to clarify. So once again, thank you. So, madam, it is over to you. Sucharita, madam. Okay.
Sir. Sir, thank you so much, sir. And the session is perfectly all right to the title, actually. Okay. And all the 9 sessions, what we have done from the day 1 to till now is a different sector. And now you have bypassed the total session, and you made us to thought perfectly interlinked to what is the mechanistic models we have to follow to do something innovative. Very perfectly aligned session, sir. Thank you so much. And it's very, very wonderful session. And you can also see a lot of accomplishments in the chat box as well, sir. Okay, thank you. Thank you for your nice words. Because I'm the last batsman, so more responsibility will be on my shoulders for this FDP. You have perfectly nailed it, sir. And thank you. Without any delay, moving on to the valedictory session, sir. Will you permit us? And I also request you to kindly stay for valedictory, sir. Okay, because sure, on the other end, we have valedictory, chief guest is on the online, sir. Okay, madam, sure. Sir, can you stop sharing the screen, sir? Okay, madam.
Happy afternoon.
Sir, very good afternoon, sir. Welcome you for the valedictory session for this 5 days eFDP program, sir.
Thank you. Thank you very much.
Hope you're doing well, sir.
Fine. Thank you. Congratulations.
Thank you so much, sir. And sir, we have the session speaker, Murthy sir. Sir is a principal of Bapatla College of Pharmacy. And Murthy sir, we have today chief guest, sir. Sir is Gopal sir. And sir is a registrar academics principal of Mother Teresa Postgraduate and Research Institute, sir, Puducherry, sir.
I know him, sir, madam. He is very well-known person.
Sir, namaste, sir. How are you, sir?
I am fine, sir.
Sir, congratulations, sir. The chat box is full of appreciations for your beautiful session.
I am also inspired about you, sir, because I also observed your lectures 1 or 2 times.
Thank you,
back,
sir.
I witnessed one of, along with you, I joined in Rajambeta, long back.
Yes, sir. Yes, sir. Annamacharya.
Annamacharya, I think 1 decade back.
Yes. You're right, sir. You're right, sir. Very good memory power.
Okay, thank you.
Sir, Gopal sir, your audio is a bit low, sir.
Low, huh?
Yes, sir.
Okay.
It's not clear.
I will come closer. Okay.
Can you connect in headphones, sir, so that it might be clear?
Okay, 1 minute.
Madam, I am audible?
You're audible, sir.
Okay. Am I audible now?
Yeah, perfectly all right, sir.
Fine.
Sir, with your permission, can we start the valedictory session, sir?
Please proceed.
Thank you, sir. And I welcome all the delegates for this valedictory session on the day 5 of this eFDP program. And I thank each one of you by name for connecting with us during these 5 days. And thank you so much for the support you have shown. And today for this valedictory session, we have a chief guest, Dr. V. Gopal, sir. Sir is a registrar academics and principal, Mother Teresa Postgraduate and Research Institute of Health Sciences, a government of Puducherry institution, Puducherry, India. Thank you so much for accepting our invite and being a chief guest for this valedictory address, sir.
Thank you.
Before proceeding further, I now request Professor Saravana Kumar, sir, to introduce the citations of Dr. V. Gopal, sir. Sir, you can.
So, good evening, all of you. Myself, Professor K. Saravana Kumar, convener of this program. It's my privilege to introduce today's chief guest, Dr. V. Gopal, sir, with your brief citations. Dr. Gopal, sir, completed his B.Pharm in Government College of Pharmacy, Bangalore, and M.Pharm from LM College of Pharmacy, Ahmedabad, Gujarat, and his PhD from Gujarat University, Ahmedabad, Gujarat. Dr. Gopal sir has received many awards, includes Bharat Sevak Award, Best Academic Administration Award in the year of 2021, Eminent Teacher Award in the year of 2020, National Best Teacher Award in the year of 2017, Siddhanta Ratna Award in the year of 2006. Sir has ex-member in AICT, NBA, PCI, and UGC profession and various bodies. Sir has nominated as member for Siddha Pharmacopoeial Committee. Sir has many professional life member of IPA, IPGA, ISTE, APTI. Sir has guided 30 PhD scholars 39 M.Pharm students. SAR has recognized supervisor for 9 prestigious universities. SAR has published 192 papers and presented 84 papers in various national and international conferences. SAR has an editorial board for various national and international journals. SAR delivered more than 300 health and moral science related programs in FM video, Doordarshan and cable TV. SAR visited many countries like Australia, Dubai, Malaysia, and Singapore for his research related activities. Now SAR working as academic registrar and principal in Mother Teresa Postgraduate and Research Institute of Health Sciences affiliated to Pondicherry University, a central university, Puducherry. So with your brief citation, I welcome you, sir. Session handover to Dr. P. Sucharita ma'am. Thank you, sir.
Thank you, sir. And I now request Dr. V. Gopal, sir, to carry for the valedictory address, sir. Sir, kindly share your valuable suggestions, sir.
Thank you. At the outset, I would like to congratulate the organizing committee.
It's not audible.
Is it okay now?
No, sir, it is not audible.
Yeah, it's audible, ma'am. So I think problem with your end.
Yes, madam, we can hear. Hello, Gopal sir, I am Sukirti Das.
Hello, madam. How are you, madam?
I am fine, sir.
Thank you, madam. Thank you. Pleasure hearing your voice. To the audience, we are very much blessed. Professor Dr. Sukriti Das, madam, a very senior member of the Central Council of the Pharmacy Council of India, is here with us. Thank you very much, madam. Thank you for your august presence.
Thank you, sir. Thank you.
Thank you. Thank you. At the outset, I would like to congratulate the organizing committee of the Seven Hills College of Pharmacy for having organized this 5-day virtual faculty development program in a very professional way. The time management, the topic selection, the selection of resource person, everything was excellent. I congratulate and thank the entire team for their excellent work. I take this opportunity to thank and congratulate the principal of Seven Hills College of Pharmacy for his efforts, for his encouragement to the team, and for his involvement and his commitment. I am extremely happy to note that a very appreciable number of faculty members have registered and have taken active part in this virtual faculty development program. I was able to witness the last speaker, and after his session, the number of appreciations that flew in the chat box itself indicates the active involvement of the participants in this beautiful faculty development program. Before COVID, the teacher was the only source of information. But after COVID, the teacher has become one of the source of information. The student is now convenient and happy with getting information from the net. So a lot of information is available. So information is not important. What is important now is knowledge. So this knowledge is given only by a faculty member, and for that a faculty member will have to update himself regularly. This updation is a part and parcel of the life of a teaching faculty. In arts and science, we have something called teacher training, whereas in pharmacy, after PG, after PhD, we directly enter into this most prestigious teaching profession. Teaching profession is the mother of all professions. And at times, we are able to see one or two teachers who act as tape recorders and photocopiers. So giving notes and just passing on the information received from our teacher to our students, then the session becomes stale. So teaching in pharmacy profession is not filling the bucket. It is lighting the fire. So the teacher, the faculty member must be capable enough to light the fire in the minds of the ignited students. The students should be made thirsty for knowledge. So once they have this thirstiness, then they will search for skills, they will equip themselves with the knowledge, and they will become useful to the society. And once if they prove their usefulness to the society, then the society will recognize the profession. Once the profession is recognized, then it will be a growth for one and all. I would like to quote an incident that took place in the life of Socrates. Socrates was given death sentence. He was given the fresh juice of hemlock, and he was ordered to drink hemlock and die. Just before he was supposed to consume hemlock, he was asked for his last wish. So the king asked him, "What is your last wish?" At the time, just outside the jail, one beggar was singing. Socrates listened to the song, and then his face became very bright. He told to the king, "I have one last wish. Please fulfill it." The king said, "Yes, it will be done." Socrates said, "I'm able to listen to a beggar who is singing outside. Please bring him in. I wanted to learn singing." Everybody present in that court, jail, including the king, were taken back. They asked Socrates, "See, this is the time you should ask for your last wish. Instead of that, you are asking that you wanted to learn singing from a beggar." He said, "See, in my life, I have learned many things, but I have not learned singing. So I cherish learning each and every day. So please bring the beggar in." So the beggar was brought in. Socrates told to the beggar, "Will you please admit me as your student and teach me singing?" The beggar agreed, and the beggar taught singing to Socrates. Socrates learned singing. He sang so beautifully that the beggar appreciated the song of Socrates. And then Socrates told to his disciples, "You will be a teacher as long as you are a student. So please remember, keep updating yourself, keep learning day by day, then only you can withstand as a teacher." Saying this, Socrates drank hemlock and he died. And that is why today in the whole world, the best teacher means it is Socrates. So the world's best teacher, Socrates, considered himself as a student and was constantly updating his knowledge. So in this, we, the faculty members, we should be updating ourselves. We should also be knowledge thirsty. The more we update ourselves, the more we will be able to discharge our responsibilities as a teacher. So in this context, Seven Hills College of Pharmacy has beautifully organized this 5-day virtual faculty development program. So I'm able to do my duty, and along with discharging my duty, I'm able to attend to a faculty development program. This is the optimum use of technology. So with the optimum use of technology, all the faculty members have been highly benefited by this faculty development program. So I once again congratulate the principal and his wonderful team of Seven Hills College of Pharmacy faculty members for organizing this faculty development program. I congratulate all the resource person, and I congratulate each and every participant who have involved themselves in this faculty development program. So we should prove our worthiness to the society. As a pharmacist, we have a long role to play. Our usefulness to the society is many a times questioned. So at that time, the service that we render to the society is very, very important. So in this context, today, knowledge is power. 40 years back, a person who had lot of land was considered rich. 30 years back, a person who had gold, silver was considered rich. 20 years back, a person who had muscle power, who was in politics, was considered rich. Today, who is the richest man? The richest man is Bill Gates. Bill Gates is not having land. Bill Gates is not having gold and silver. Bill Gates is not in politics and is not having muscle power. Bill Gates is having knowledge. So knowledge is power. So this is a knowledge era. And during the COVID period, the people who are capable of delivering a vaccine of good quality at the right time, that knowledge made them powerful. That knowledge made them useful to the society and community, and their services were acknowledged. So knowledge is power. So let us update our knowledge, and in this process, the role played by Seven Hills College of Pharmacy is always appreciated and recorded in the history of time. The participation of the teaching faculty in this faculty development program again needs appreciation at all corners. Amidst their busy schedule, they have taken active part and they have updated their knowledge. Congratulations to all the participants, and together we shall have more such faculty development programs regularly. Keep ourselves updated and be beneficial to the society and make this society a better place to live in. Thank you. Thank you for this nice opportunity. Thank you. Thank you, one and all.
Thank you, sir. Nice.
Thank you, sir. Thank you so very much. And you really hammered our brains and trying to infuse what is the responsibilities of teachers. Still, we need to do in the area of NEP actually. Thank you so much, sir. And I now request Gopala Krishnamurthy, sir. Can you please add few inputs in this valedictory address, sir?
Actually, I am not having any opportunity to share because all the things was expressed by Gopal sir. So, so Gopal sir explained what is the duties or need of faculty development program and how you conducted, how such kind of programs are useful to the students. So most of the message was well covered, delivered by respected Gopal sir. So on behalf of me, once again, I am thankful to the organizing committee and participants. My wholehearted wishes to all faculty members, and you listen the words expressed by the senior professor and try to update your knowledge. As long as we are in the profession, definitely we have to update our knowledge. And I hope you will follow the suggestions, message given by Gopal sir. Thank you, one and all.
Thank you. Thank you, sir.
Thank you, sir. Thank you so much. And Gopal sir, I now request you to kindly do the felicitation for Gopala Krishnamurthy sir, and kindly do the honors, sir. We'll be sharing the certificate.
Sure, sure. Yeah. Can the certificate be in slideshow mode? Yeah, thank you. On behalf of the Seven Hills College of Pharmacy in Tirupati, the most sacred place in the earth, on behalf of the organizing committee of the 5-day faculty development program, shortly called as eFDP, on a theme of the current thirst area, pharmaceutical innovations and protection strategies, it is my pleasure and privilege to read the certificate of appreciation. This certificate is awarded to Dr. T.E.G.K. Murthy sir, the professor and principal of Bapatla College of Pharmacy, Bapatla, Andhra Pradesh, India, in recognition of his contribution as a resource speaker for the title Mechanistic Models in Pharmaceutical Innovations. So the title is itself very innovative, and he has made the participants understand, observe, and get involved in this beautiful session. So his involvement in this 5-day faculty development program with the theme pharmaceutical innovations and protection strategies is much appreciated. On behalf of all the participants, I thank him and I wish him all the best. My prayers to the Almighty to shower his blessings on this resource person so that he could pass on this valuable information to all the needy faculty members. Thank you very much, sir, and please accept my appreciations.
Thank you, sir. Thank you very much.
Thank you, sir. Thank you very much.
Thank you, sir. It's very, very wonderful. And I'll take a few minutes of time to listen the valuable feedback from all the delegates. So, the participants are welcome for the feedback. I request all the participants to please raise the hand in the chat box so that we can unmute you. Kindly spare a minute of time with us. Yes, I can read a lot of messages in the chat box, but still.
Ma'am, the e-certificate will be sent through emails to all participants.
Yes, within a week from CSIR, we'll be sending all the certificates to the delegates.
Okay, thank you.
Thank you, sir.
Excuse me, can I speak, ma'am?
Ma'am, please. Good afternoon. Welcome you, ma'am.
I'm Sukirti Das, Professor, NITIS College of Institute of Pharmacy, which is in Assam. Now, I sincerely attended all the sessions for this 5 days FDP program, and I am really in all praise for everybody. The management, the principal, the organizing team, all of you have done a wonderful job. And I am really, it is true to the words that I have upgraded my knowledge by attending this program. So, and I am also, in addition to that, I was very happy to just see Gopal sir and hear his wonderful words after many days. At the same time, I would like to congratulate the management, as I have already told, and personally, I am really benefited, and I can assure that everybody has found this FDP wonderful, unique, informative, and the resource persons were really, really very knowledgeable, and they have done best to make the say participants understand the topic. And thank you for giving us the opportunity to attend this FDP. And I hope in future also we will be getting the chance to attend the same. Thank you. Thanks a lot.
Thank you, Sukriti ma'am. Your words are really a driving force for us to do the best in next coming FDPs. Any other participant? Surya Prakash Reddy, sir. Good afternoon.
So with your permission, I would like to share my feedback. So I am Dr. Surya Prakash Reddy, Professor in Annamacharya College of Pharmacy. So very good evening to Niranjan Babu Garu, Principal, 7 Hills of Pharmacy, TGK Murthy Garu, Gopal sir, Convener Sharan Kumar sir, and all coordinators and all dear faculty participants. So indeed, the theme selected is the need of the day. And I would like to congratulate the convener and all coordinators for organizing such a great team-oriented eFDP for 5 days. I know the pain in conducting such a beautiful program. So it took all the pain to make it a grand success. And blend of the talks are exceptional. And the time management is awesome, really. So 10 o'clock means exactly by 10 o'clock, the sessions will start. So the time management is very awesome. And all the speakers delivered the talks to their best. There is no doubt in this. And hope in future you may come with yet another exceptionally great team-oriented FDPs, hopefully in physical mode. So all the best. And Through this FDP, I learned a lot of things which are not known to me. Thank you. Thank you for organizing such a great, wonderful event.
Thank you, sir, for your valuable feedback. And any other participant is willing to give the valuable feedback, kindly raise your hands.
Hello.
Yeah, madam, please.
Hello, Sucharita ma'am.
Yeah,
good evening,
afternoon,
ma'am.
Yeah, good evening, ma'am.
Good evening, ma'am. So, good evening to one and all. First of all, I thanks to our principal of Seven Hills College of Pharmacy, Dr. M. Niranjan Babu Garu, and Vice Principal of Seven Hills College of Pharmacy, Sumlata Madam Garu, and special thanks to our convener of Seven Hills College of Pharmacy, this 5-day virtual FDP program, Dr. K. Sarvanakumar Garu, and I would like to congratulate. Congratulate our coordinators this program. So here I sincerely attended in this 5-day FDP program. Next one is I would like to share my experience and feelings in front of you. I can gain knowledge and different areas like commercialization of the pharmaceutical innovations, and not only commercialization of the pharmaceutical innovations and intellectual properties in different case studies. So pharmaceutical research protections. Next one is in regarding to IPR. So patenting with different strategies, examples, and very innovative session, that is mechanistic model in pharmaceutical innovations. So very, very wonderful session. And especially I am very happy to for selection of a title and organizing of this program, and selection of the eminent speakers also. Very good peoples are there. So finally, well organizing and with very coordination is very good. So finally, I would like to congratulate the who are involved in this program. So thanks for giving opportunity in front of you. Thank you so much, ma'am. Thanks for giving opportunity once again. Thank you so much. It is very informative session and very, very wonderful session. So thank you so much for this opportunity. Thank you, ma'am.
Thank you so very much, ma'am, for sharing your valuable feedback, and your points are noted. And now I request Professor Pushpakumari Madam to kindly felicitate today's chief guest for the valedictory session, Dr. V. Gopal sir. Madam, on this occasion and on behalf of Principal sir and the management and the team of Seven Hills College of Pharmacy, I request you to kindly felicitate today's inaugural valedictory chief guest. Kindly do the honors, ma'am.
Thank you, Dr. Sucharita. Namaste, sir. So on behalf of Seven Hills College of Pharmacy, Autonomous Tirupati, management, principal sir, and the organizing team of this 5-day eFDP program, and all the other teaching faculty of Seven Hills College of Pharmacy, it's my honor. It's my honor, sir. I kindly request you to accept this small token of gratitude in the form of felicitation. So this certificate is awarded to Professor D.V. Gopal sir, Registrar, Academics and Principal, Mother Teresa Postgraduate and Research Institute of Health Sciences, Puducherry, as a chief guest for valedictory function in a 5-day faculty development program on pharmaceutical innovations and protection strategies conducted from 18th to 22nd July 2023, organized by Seven Hills College of Pharmacy, Autonomous Tirupati, Andhra Pradesh, India, in collaboration with CSIR Institute of Minerals and Materials Technology, Bhubaneswar, India. Sir, kindly accept this, sir. This is a token of gratitude from our side.
Thank you very much. Thanks a lot. Regards and best wishes.
Sir, I am a teacher of Sudhir Kumar, sir, who is working in here. Yes, sir.
Yeah, great.
Yes,
Pleasure.
sir. He was
meeting
my
you.
student in M.Pharm pharmacology, sir.
Oh, excellent.
Yes, sir. I heard about you very much from him, sir. So in that way, I know you very well, sir. Thank you, sir.
Thank you very much. Thank you.
Yes, thank you, sir. Thank you so much,
Madam. And moving ahead, I now request the organizing secretary of this virtual 5-day FDP program, Mr. Mallikarjuna sir, to present oath of thanks.
Thank you, Dr. Sucharita, ma'am.
Yeah, we know
So very good evening to the honorable chief guest, respected principal, Dr. M. Niranjan Babu garu, colleagues, and my dear delegates. So myself, G. Mallikarjuna, Associate Professor, Department of Pharmacology, on behalf of Seven Hills College of Pharmacy and its entire fraternity. It's an immense pleasure for me to propose the vote of thanks. So this is one of the momentous day for us, as SHCP has organized a 5-day virtual faculty development program on pharmaceutical innovations and production strategies in collaboration with the CSRI, Institute of Minerals and Material Technology, Bhubaneswar, India, from 18th July to 22nd July 23. And my sincere thanks to the Honorable Chief Guest, Dr. V. Gopal Sir, Registrar Academics and Principal, Mother Teresa Postgraduate and Research Institute of Health Sciences, Puducherry, for taking time from his busy schedule to be with us and our delegates. Sir, your words and messages on this special day is highly appreciated, sir. Thank you very much, sir. And I feel incredibly honored and privileged to extend my sincere word of thanks to the Council of Scientific and Industrial Research, Institute of Minerals and Material Technology, Bhubaneswar, India, for their unflinching support and collaboration for organizing such a wonderful program. And I especially thank Dr. T. Pavan Kumar, sir, Senior Scientist, CSIR, Institute of Minerals and Material Technology, Bhubaneswar, India, for his enormous support in initiating this 5 days virtual faculty development program. And your support will always be remembered and honored, sir. We hope and wish to have more support for organizing such wonderful programs in the future. Thank you, sir. And I would like to take an opportunity to express my appreciation to all the resource speakers of the 5 days FDP sessions for their presence and making this program a successful one. I would like to thank all the faculty delegates from the universities and all the institutes all over the India for their registrations to the program, so which has reached nearly 2,400. So very special thanks to all the faculty delegates who have made this program a successful one. And a special thanks which goes to Dr. M. Niranjan Babu garu, Principal, and Srimati M. Sumlata garu, Correspondent, Seven Hills College of Pharmacy, so for their unwavering support in making this virtual faculty development program a grand and successful one. And I would like to express my sincere thanks to the convener of the program, Professor K. Sarvanakumar garu, for organizing such a wonderful program. Thank you, sir. And I must also acknowledge the efforts and the teamwork of the organizing committee, Seven Hills College of Pharmacy, to making this FDP a grand and successful one. And I extend my sincere thanks to Dr. E. Sunil Kumar, Associate Professor, Department of Pharmacy Practice, who led this virtual FDP uninterruptedly. And I express my thanks to all the teaching and non-teaching staff for their support for making this program a successful one. So the wheels began rolling a month ago. So when this FDP was initiated, since then, everyone became the part of this FDP in many ways. So my sincere thanks to everyone who has supported this program to make a grand one and successful one. So we feel enlightened and believe that we got to learn many things from this program. The words would fall very short of describing how much knowledge we could derive from this faculty development program and the important networks we could create. So with these warm words, we move to the end of 5 days virtual faculty development program. And thank you very much to everyone for giving me this opportunity to propose the vote of thanks on this day. Thank you. Over to Sucharita, madam.
Thank you so much, sir. And the objectives of this FDP program is fulfilled, and I hope everybody will share the knowledge to students as well. That is the objective of organizing this FDP. So now, with due permission of the chief guest for this valedictory program, sir, can we go for national anthem? Yes, sir. We can curtain down officially with national anthem, sir. Thank you.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, sir. Thank you so much. Thank you all the delegates for joining into this 5 days FDP program. Feedback links are shared in the Zoom chat box and also YouTube description box. You can find that, and the e-certificates will be provided to you within a week. Thank you, sir. Thank you.